has some seed business, which is like its secondary activity, and we hold around 10% market share in Croatia as well as in neighboring countries, and some 5% market share in Turkey. So the global seed market can generally be separated into two tiers. The first tier is only 10 largest companies, 3 of which are the Corteva, the Bayer, and I can't remember which is the third one. I think it's Syngenta, which all reported the sales of seeds of more than $30 billion only for 2018. And in the other tier are the 31% of the rest of the stakeholders of the market. So of the global seed market, only maize holds 42%, and all the other crops are hold the rest. So it's nearly $18 billion US. This is the information ordered by the FAO, made by this agency in 2019. So when I say tangled crossing scheme, so there is actually a cheat sheet for exploitation of maize heterosis, heterotic effects, and this is called heterotic groups. So heterotic groups, as you can see here, just a second, so I turn on my laser pointer. So you can see there are generally 4 heterotic groups that can be crossed, lines from which can be crossed, or among which can be crossed. So among stiff stalks and iodines, you could expect some predictable and considerable amount of heterosis, as well as between any other group and the representatives of the other group. So in theory, I say in theory, there are only 4 heterotic groups in maize, but this is not the case, because you can combine like 3 of the heterotic groups or 2 of the heterotic groups on one side and develop lines or progenies in that manner, and then you can cross them to like some purebred Lancasters and also get some predictable and considerable amount of heterotic effects. So that's why I say in theory, because these are very arbitrary interpretations of this is basically a cheat sheet. So in practice, this is what it looks like. These are the results of genotyping using the 50K SNP chip from Illumina of all of our gene bank or our old breeding program. So here are Lancasters, here are the stiff stalk synthetics, here are the iodines, and you can see Ohio germplasm, which is like in this scatter plot, also separated from Lancaster pool. So as I said, you can blend these inbred lines, and each breeding program is actually tailored for the target population of environments, and the target population of environments is basically the environments in which you wish your product to perform the best. So basically, if you have some temperate environments, you will tailor your breeding program for temperate environments. If you have some environments with expected loads of heat, you will tailor your breeding program in such manner. So in reality, this is what it looks like. Basically, your breeding program is only a blend of inbreds tailored for the target population of environments of the company performing the breeding. So when you have many generations of breeding, this is what you would expect. So basically, you cross some 2 inbreds. This is only like very dull example, but you have some mean value and you have some cutoff of, let's say, some tangible 10 to 15% of inbreds, and then you perform the cutoff from generation F2 to let's say F5, and this is what you are expected to get. You should get some increase in mean. You should have some variance, and but this is what your afterwards what your breeding program looks like, and this is what you end up with. So basically, the question in plant breeding community today is this resilient to climate change? So if you have narrowed your the diversity of your breeding program, is it really only adaptationally fit for your target population of environments, and what if this target population of environments changes in any manner. And this is where we come to the genetic resources. So genetic resources basically represent the gene banks in which the accessions are stored, which were already utilized for human benefits. So this is not the utilization, this is actually reutilization or resurrection of these lines. Inbred populations, open pollinated varieties, and so on. So basically, the first commercial breeds in maize were the open pollinated varieties. This was like during the 18th and 19th centuries in mostly in US. And these were followed by double-crossed hybrids during the beginning of 20th century. So today, more than 95% of the breeding market is covered by single-crossed hybrids. And you can see that there is a vast amount of diversity that has been cut through the breeding throughout the years to only several player inbred lines, let's say, let's call that. So you can trace all the modern inbred lines covering most of the breeding in maize breeding, commercial maize breeding in the world to only several historic US open pollinated varieties from the end of 19th century. So when we say genetic resources, first thing I think is that this is really obsolete, and these are like some accessions that flower early, that are really obsolete in terms of agronomic performance, have very low yield and poor phenotypic performance. But and if you would like try to go to the pre-breeding again with these open pollinated varieties, you would end up with having many lethal alleles, many very poor progenies that would come out of these varieties. So there is also the problem with linkage drag, which is, I guess, in breeding the worst kind of problem. And what it means is this, actually. If you have some loci, let's say some unfavorable loci that is not so close in terms of genetic distance to the some loci causing some favorable agronomic performance, you don't have problem, because this is the linkage that can be easily broken. But if your loci are close in some, let's say, cold spots of the genome, and there is low probability of recombination between these two loci, you will end up with many unfavorable loci covering the effects, masking the effects of favorable loci, and basically you would have, these progenies would end up in trash. So this is where we come to some new technologies. So this is what the whole genome technologies offer, whole genome sequencing technologies. So you have total genomic saturation, you have high resolution, and also to screen the diversity, you have a large number of copy number variants and presence absence variations, which you can screen for, of course. So within this grant, the Center of Excellence in Biodiversity and Molecular Breeding, we genotyped more than 800 inbred lines, which were stored in gene bank of former Yugoslavia in Maize Research Institute Zemun Polje, which is under the ingerence of Dr. Violeta Anđelković. And these are mostly lines from Southeastern Europe, but there are also some from Russia, Poland, ex-DDR, ex-Czechoslovakia, People's Republic of Croatia, of China, and as well from Korea, Brazil, Mexico, and so on. So we genotype these accessions, which are inbred lines, which were bred from locally adapted open pollinated varieties. We genotype them using the Axiom 600K SNP platform, which gave us the resolution of some 600,000 SNPs and some 16,000 of presence absence variations and copy number variants. And generally, for the first step, we screened for some signature of selection. And when I say signatures of selection, basically if you perform selection for some favorable loci, what you would expect due to the linkage, you will get some signals near this loci, and you will get the local loss of diversity. So this is not maybe the happiest solution to screen for signatures of selection, but this is very informative in terms of what was done with this genetic resource, because we found a funny signal like this, which is for flowering promotion factor like 1 protein. This protein causes earlier flowering in maize. So basically, this is what this is actually the detection of work done in a wrong way during the breeding, because workers were selecting and selfing only the earliest progenies and basically fixed this allele, right? So there are also some other signals. These are changes in linkages equilibrium patterns, shifts in site frequency spectra, and local losses of variability, which are also screened. This will probably be published, and I hope this will be in consideration in theoretical and applied genetics by the end of this month. So currently you can check it out on bioRxiv. So we performed some other scans for local loss of variation and shifts in site frequency spectra and linkages equilibrium, and we found some very interesting signals. When I say interesting signals, we found even on chromosome 5, we found selected loci that should be increasing yield, grain yield in maize test crosses in some temperate European environments with cold nights. And this was published in Plant Physiology Journal by group from INRA in France and Wageningen from Netherlands. So this is something we are really proud of, because this is a really inspirational paper. I suggest you to read it if you haven't already. And there are also many more signals that were detected that are very interesting adaptation-wise. So generally, this is the first step towards the reutilization of this large gene bank and these great genotyping results. And these results can be utilized for like QTL libraries, targeted recombinations, transformations, and of course to enrich the germplasm for newly released varieties with old diversity. And I guess there are some prospects for this germplasm adaptation-wise, because where we are currently in maize breeding and in breeding general, it's questionable. Is it smart place adaptation-wise? So basically, that's all from me. I hope that you have at least some got at least some perspective on genetic resources and what they can be utilized for. Thank you.
Thank you very much, dear Galić.
And if there are some questions,
yes, if anyone has a question, they can ask now. I think there is no question.
Well, I have a quick question. Interesting talk, thanks. I was wondering if the heterozygous groups you showed in the beginning form some coherent geographical distribution or not?
Yeah, of course. Basically, when we talk about stiff stalks and Lancasters, these are the later maturity inbred lines, which were made from some populations from the from equatorial area of Mexico. And when we talk about iodines, these are basically inbred lines that are straining from reed yellow dent program from 19th century, which was held I think somewhere north. I'm not sure. More north. I think it's from Iowa. So generally, yeah, there is a pattern.
Mhm. Good.
Yeah, I was going to ask if you are considering using double haploid technology in your work to sort of quickly also breed and dissect different alleles and different SNPs and what they're doing.
Yes, we have, and there are some great results regarding the OPVs and double haploids from Albrecht Melkinger at University of Hohenheim. So generally, yeah, we were considering this, and this is probably the next step. But what I was showing now is the gene bank of inbred lines. So these are already purebred inbred lines in total inbreeding. So there's no need to induce haploids from them. But generally, to screen for diversity in open pollinated varieties, yeah, double haploids would probably be the best. Although some new results suggest that there is some skewness in induction of haploidy. And there is some ascertainment bias towards some variants compared to the others.
Right.
So I hope.
And I guess with these varieties, again, they're OP varieties that are quite old, some of these. But did you see some residual heterozygosity still in some of these lines over time?
Yeah, actually there is, like, depending on the inbred lines, there is residual heterozygosity in terms of like from 0.5 to 5%.
Okay.
Yeah.
Okay.
I have another question, if nobody's.
Okay, you can ask.
I was wondering, in your breeding programs, if you came across a lot of linkage drag, or is it exceptional?
Linkage drag. Well, linkage drag is mostly the problem when you take the inbred lines out of the OPVs. So generally, if you have like some open pollinated variety or some old cultivar, what you would expect that most of the poor agronomic performance will be due to the linkage drag, and you risk basically cutting off the progenies that maybe hold some favorable diversity, but is masked due to the linkage drag. So that's general issue with breeding without genotyping prior to pre-breeding, let's say.
I want to ask a question also.
Sure.
Yeah, thank you, Vlatko. This is nice presentation.
Thanks.
As you highlighted in your presentation, you said the development of climate resilient cultivars is getting the top priority among all the breeding community. So as a maize breeder, could you say what kind of traits or how you can describe the idiotypes of resilient maize cultivars?
Well, that's really tangled because there are some my subjective opinions on it, but generally there is only one trait that forms the idiotype, and is the grain yield. So from like perspective of morphophysiology, I would say that you have to have cultivars that are more prone to the high vapor pressure deficits. You have to have cultivars that can tolerate like some higher density of higher planting densities. But generally, the only idiotype is defined by the grain yield. As one famous breeder said, 3 traits are most important in maize breeding, and it's the grain yield, and grain yield.
Okay, thank you.
Yeah. Thanks. So if that's all, I can stop sharing.
Are you familiar with the short statured corn that Bayer Monsanto are coming out with, and some of the results there? Because that they're able to increase their planting density, and it seems to still have a good yield. At least that's what their YouTube advertisements and all talk about.
Well, generally with Monsanto hybrids, they are quite distinct in field, and you can really say, okay, that's the decalb. It's the decalb.
Yes, originally, yes.
So they're really distinct in appearance. So generally, when you see it, they are lower in stature. They are less leafy, and you would expect them to yield less, but that's not the case. So yeah, I am familiar with that. Yeah, it's a great effort, I guess. The only problem, but that's the problem of whole scientific paradigm, is that in academia you chase some findings that are already discovered in some private companies. I guess that's just my opinion.
Although there's often a lot of things in academics that we find interesting too. So I think that goes both ways, quite honestly.
Yeah, sure.
Yeah.
Okay. Thanks for your questions and discussion and invitation. So I will stop sharing now, if that's okay.
Okay, Vlatko, thank you very much for your contributions.
Thank you.
Then now we can move on to another speaker, Mr. Abdullah Barakat. Dear Abdullah Barakat from Morocco,
Go on.
is currently working in the United Kingdom at the Jamet Hutton Institute as a plant molecular geneticist, and he will speak about meiotic recombination and gene editing on barley. Dear participants, Mr. Barakat will answer your questions after finishing his speech. You can write your questions to the chat. Welcome, Mr. Barakat. If you're ready, you can start.
Thank you. Am I okay for time? About 35, 40 minutes? 35, I would say. Yeah.
Okay.
So I'll share my presentation.
Okay.
Okay. Can you see it?
Yeah, we can see.
Okay. All right. I would like to thank all the attendees. I hope you are all well and safe. I would like to thank particularly the organizing committee for inviting me. And it's good you heard the first talk, which basically gives you the start and the end of a breeding program where you cross plants, and in a black box of meiotic cells, the arrangement of new allelic are sorted out, and that's what you get at the end. My talk will be focusing actually about the mechanistic side of the meiosis, what happens inside this black box, how genes are segregated, and how can we manipulate that process once we understand it. So, I don't know, can you see my arrow? So basically, all the work that I will talk about today has been published mostly, some in preparation, and you can find in my website here of the Institute a list of those publications. There is also a link to 2 videos, one regarding gene editing with CRISPR, and one on gene shuffling during meiosis. We call it gene shuffling because it was funded by EU, and the name of the project was Shuffle Project. Before I start talking about science, I would like to let you know where I'm based. This is Dundee. This is a Tay River. It's a lovely place. And actually, it's quite nice because we have a microclimate in this area. Quite often, we don't get snow, and only a mile, 2 miles away, and you find snow, for example. And I started at the University of Dundee at this and here for about 18 years. But actually, our plant science division was moved to what was called then Scottish Crop Research Institute. It's about 3 miles from the center. This picture is a little bit old. Now the institute is actually called the Jamet Hutton Institute, which is merged with an institute in Aberdeen in North. And actually there is a lot of development has been done in the riverside where basically nowadays if you come to visit, you can visit the famous V&A museum. It's lovely to see. It's fantastic. And also locally, we have now development underway of the what we call international barley hub that's going on this site here. But also we are really happy that we have one of the first station of vertical farming in Scotland, and it's fully automated and it's really great. You can find it online and you can see what you do. Everything is automated with LED lights and nutrient and so on. It's great. Now I can talk to you about basically why do we want to study meiosis in barley. The reason is that, as you know, meiosis is a shuffling of genes, and it allows chromosomal integrity between the parent chromosomal to their children, basically. And also because it makes new assortment, you get opportunities for new selection, new evolution of new varieties, for example, and so on, and new species. And as you know, meiosis is really important in terms of health and fertility in human, and it's important for securing our food given the global warming and also an increase of population. We use barley because it's it's a diploid plant. It's a nice model for crops, and also economically it's important for UK, especially in Scotland. It's used as a crop for whiskey distillery, for example. But quite often plant breeders have difficulty, as previous talk showed with linkage drug, for example. They have difficulty creating new varieties with the traits that you want. And why is that? This is because recombination in barley or rice in general tend to happen at the end of the chromosomes. This is a single myocyte, a single nucleus that contains 7 pairs of chromosomes. The pairs of chromosomes, it's from the mother and the father, and they are linked only at the ends, but hardly ever in the middle. And in the middle, towards the Central America areas, there are about 30% of the genes, but they're stable, rarely recombined, and we would like to tap into that reservoir in a way. So the aim of the project of the last 5 years is to understand the mechanism of meiosis in order to manipulate the crossover frequency and the distribution across the chromosomes. So in barley, this is a spike at the age of 6 weeks of growing plants. Each spike contains multiple floret. Each floret will make 1 seed, and basically this floret has, if you dissect it, 3 anthers and the ovule that will make the seed, basically. If you look at it using a transgenic plant that has GFP, it's a green fluorescent protein from jellyfish, and you can express it in meiotic tissue, and you can see hopefully the anthers here, and here it's a bit like a fish stripped of flesh, basically. But if you make a section of this anther, what you get is 1, 2, 3, 4 locules where the pollen develops here and here. Those pollen mother cells undergo meiosis that I'm going to talk to make mature pollen. Now, at this stage of meiosis, you can literally squeeze the myocytes out and they come out like a sack. I call it, in the lab, we call it caviar. And basically, you can stain these cells, and each dot here is a nucleus that contains these chromosomes that are paired, basically father and mother chromosomes. And we know using DNA probes specific to different chromosomes, we know which one is chromosome 1 and 4 and so on. There are 7 chromosomes in barley. As you can see, again, they are linked mainly at the ends like a donut. And since there are 7 chromosomes, we tend to find cells with 14 crossovers. But some cells have less, some cells have a lot more. What we want is to actually shift this distribution towards this end to have more crossovers and everywhere, not just at the end. This diagram basically draws this blob basically of chromosomes here, showing the centromeres, and they are recombining at the ends at the telomere. What we found, what the researchers in meiosis found, is that actually you can split these chromosomes into 3 zones. Zone 3, which is centromeric, where there is no recombination or hardly any, and the end is zone 1 here and here, and zone 2 where recombination happened. So what we want is to shift this recombination here towards the end, towards the center. Don't worry, this is to show you how we follow meiosis in cytologically under the microscope. So this is a single nucleus that looks like a plate of spaghetti. These are chromosomes that condense and undergo meiosis, different configuration, and the end in the metaphase 1 that I showed you earlier. We have probes, antibodies that recognize specific protein, like this one called ASY1, ZIP1. Don't worry, I will come back to that, show you how they work. And we can label them with different fluorescent markers, secondary antibodies. And what happens is you can follow both of them. The blue is actually the chemical we use for staining DNA. So you can see that's why it's in the nucleus, and the proteins, the chromosomes change configuration, and amazingly at the end they are actually separated. If anything goes wrong, the cell will die, and you will get sterile plants. A number of genes are involved in this mechanism. Only few are shown here. So what happens just before meiosis starts, the DNA is duplicated, like you make a photocopy of a book, and you get, for each chromosome, its sister chromatid. And this enzyme here, with others, they introduce breaks. There are about 500 breaks that are introduced in each cell. And those breaks have to be repaired, otherwise the cell dies. And I'm not going to go through the details. There are proteins that are against recombination, some helicases. There are proteins that allow recombination to happen. So it's the balance between these proteins that end up generating only 20 crossovers out of 500, and the rest is called non-crossover, where basically this chromosome, the homologue, it's used just as a information copied into that, and that's it. They don't exchange completely. In reality, actually, these structures you see here, they are, if you look in 3D, this is basically the sister chromatids that I showed here. Yeah, replication, that's one chromosome. And the second homologue is here with its chromatids, arranged in loops, basically, to pack in as much DNA as possible. And those homologues are linked by proteins of the what we call synaptonominal complex. It's amazing structure, and I'll come to that later. In addition to this configuration, actually the DNA is wrapped around nucleosome, which to form a nucleosome with histones. These histones undergo chemical changes we call epigenetic marks that are important to tell the cell, yes, you can have a crossover here or no, and so on. They're important in gene expression and so on. So I'm going to take you step by step from DNA double helix to all the way how we make those structures. So this is the double-stranded DNA, which is wrapped around histones to make basically nucleosome. Each nucleosome attached to the other one, you end up with like beads, okay? And if you look at the electron microscopy, that's what you see here on the top. Then these beads are arranged into what we call fibers. Those fibers, or chromatin fibers, actually look thicker in the electron microscopy. As I said before, you get 2 chromatid sisters per chromosome, okay? So that's the replication. They are held together by proteins called cohesin, and those proteins I mentioned in the axial element that zip them together. So that's how they look again. Those proteins, cohesin, that's how they wrap the fibers. And what happened, actually, the chromosome is not linear. It makes all these loops here so that it can pack as much DNA as possible in a small space of the nucleus. And this is one of the protein here in the axial element that basically links them. And the 2 homologs come together starting at the telomere where they are aligned basically. And if you use antibodies specific to this protein, that's what you get. You use another antibody, then you see how they are zipped together. So this is a wild type, for example, and it can go wrong or different in the mutant, basically. This is, again, the protein that I mentioned called ZIP1. And the ZIP1 I highlighted Y because in the literature you find ZIP with Y or E or I. I don't know why, but that's the way it is. Okay, so the chromosomes are zipped all the way long, basically, and this is what you get in the wild type, in the zygote stage. In the mutant, somehow it's not happening. Yeah, this zip protein is not aligning properly. And then what happened is at the end of, towards the end of the meiosis, this ZIP1 and AS1 should be removed so that the chromosome can separate. So the mutant, somehow those proteins are not separating properly. That's why the plant becomes a bit of a mess, basically. And towards the end, that's what you get in the mutant. These proteins are staying sticky. They don't want to move out. And because of that, in the wild type, you get nice donuts here, 7, but in the mutants, there are 3 cells from the same, showing all kind of linkage and basically all these breaches will make it difficult to segregate chromosomes properly, and the cell will probably die. So I showed you earlier that we can count those chiasmata, but to be honest, I'm not a cytologist, but I find it quite subjective. But we can use probes of antibodies that recognize proteins that are specific to those crossovers, like this one, MLH3. There is another one called MLH1. And doing the same count, well, we find exactly the same, that actually there are more crossovers at the end here than in the middle. Okay, we can even find out where those crossovers happen using antibodies that recognize the methylation I mentioned earlier of the histone, for example. And what we found is those crossovers tend to happen in the euchromatin, where the chromatin is open, loose, and it's expressed, not in the heterochromatin, which is full of transposon element, repetitive element, and basically it has to be protected, otherwise things will go badly. Overall, by studying genome-wise sequencing that previous talk mentioned, well, again, it confirms that there are 3 regions of, in terms of recombination, the middle, which is the desert of recombination, and the recombination tends to be increased at the end, basically. And that's where there is a high evolution of genes, while here they are quite stable. As mentioned previously, also we use 50K array. This is an array that has 50,000 detection SNPs, basically. So we can scan when you cross 2 cultivars, for example, we can follow those SNPs from one individual to another, from one generation to another, and we determine where the recombination actually occurred and so on. So our aim was to really disrupt this recombination pattern and increase it towards the middle. So what we did is we have in barley a whole set of genomics. We have mutants, natural and induced mutants. We have all the genome sequencing and transcriptome and all that stuff. We performed transcriptomic and proteomic experiments using anthers of barley at meiotic stages. That work is published, so you can look at the paper. There are also lots of papers on meiotic genes in different species, not just barley. So using that knowledge, we actually selected some candidate gene, and we use our toolbox of gene validation, basically. So if you have a gene of interest, I'm sorry I'm explaining all this because I'm aware there are a lot of students here. So if you have a gene of interest, that gene can make mRNA to translate it into protein that can have post-translation modification like phosphorylation that result in a phenotype. Any gene is regulated with small RNA or transcription factor and so on. What we can do is we can insert the same gene or a different gene as what we call a transgene. This transgene can be designed so that it makes a protein. This protein can be the same as this one or different with fluorescent markers, for example, like GFP and so on. We can even introduce post-translation modification or change them so that they don't work. And then we assess the phenotype of that. We can also design this transgene to make aberrant RNA, which is not good for protein. But with this, it will trigger what we call RNAi, which is gene silencing of the endogenous gene. Ultimately, what we want is to be able to knock out or modify the gene itself. And we have, as I said, mutant, but that's not enough sometimes. We can use chemical mutagenesis that changes, but that's really, I will talk about that, it's quite random. You cannot control it. But nowadays, we can actually do gene targeting using things like CRISPR-Cas9 that targets that gene, nothing else in the genome. And that's really powerful. And once we have our mutant, we can then use immunocytology that I showed before. We use genomics and so on to characterize the effect of the mutation on meiosis. Is it good or bad, and so on. So before I get to that, I want to explain the principle of gene editing, which is basically inserting or deleting or replacing your gene of interest and nothing in a specific manner in the genome. It's more precise, more efficient, and simpler than conventional GM method. GM methods, you make a transgene, you insert it, it goes more or less randomly, and then it's quite often not stable from generation to generation. Gene targeting is more powerful. And there are a lot of tools that have been created. It was frustrating, but now with CRISPR, which is the most popular one, we can really change an awful lot of things in the genome in a precise manner. So, as I said, the main thing is CRISPR and other nuclease can introduce a break where you want here or wherever. And that's the problem solved, because without that, you can't do anything. And actually, the first nuclease, artificial nuclease that was made, it was made using a transcriptional factor binding domain, zinc finger domain, fused to a bacterial enzyme or restriction enzyme, just the domain that like scissors. And but this was very complicated. It takes 2-3 years to design those proteins, so it's quite frustrating. And then another new one was designed using a transcription activator, like from a pathogen of plants again. And that was a lot better, but still quite challenging. And finally, this bacterial system was once understood, redesigned so that we can use it in plants and animals. And it's used now in medical sciences and in agriculture, and it's just amazing. And we have applied now this tool in tobacco plants and Arabidopsis as a model, but also we translate some of our finding in potato and in barley. Again, areas that we are working on is mainly recombination, both in somatic and meiotic cell, but also cell wall, like I mentioned, lignin biosynthesis, like for biofuel, and also like in potato, food quality, and barley, quality of seeds, and so on. So CRISPR-Cas9 stands for clustered, regularly interspaced, short palindromic repeats. What this system came from, it's an adaptive immune system of bacteria. Not all bacteria have it, but some species. And what happened, people knew about it since early 80s, but actually they thought it was an artifact. It's only lately that once it's understood what it is, it's a system basically that detects viruses of bacteria or plasmids from other strains. And when they are invading the cell will capture a bit of their DNA and put it in an array, kind of like an archive. And next round, when the virus comes back, these archives are expressed, and if it's the same, it will be targeted and chopped. That's basically why it's adaptive immune system. And as I said, it was repurposed by scientists, and all what you need is this protein called Cas9 and a guide RNA, which has this structure that's recognized by the protein, and you can add 20 base pair that are homolog to your gene of interest. And that's it, really. So simple. We made vectors for both monocot and dicot plants in a single ligation. You just add those 20 base pair and you're ready. Once this CRISPR-Cas complex, this Cas9 guide RNA complex breaks your DNA of interest, it is often repaired by the cell, plant or animal, by this pathway of non-homologous end joining. What it does, quite often it's in a perfect way, but sometimes it introduces mistakes, it makes mistakes. Those mistakes can generate knockout of your gene, and that's what we screen for. Occasionally, very rarely, you can actually do replacement. If you have a copy of a gene that you want to insert, then it can be done. But in plants, it's not very efficient yet, but it's just a matter of time. How do we introduce these things into plants? You have everything ready. How do you put it in plant cell? We tend to use agrobacterium. It's a natural engineer, basically. These are those bacteria and the plant cell material. What you do is they inject the DNA and it makes these galls, and they can be massive in nature. So it's natural way of doing. things, but we can also have viral vectors. We can use naked DNA. We can use RNA, Cas9 and guide RNA as RNA. We can also use the Cas9 protein forming a complex with the guide RNA. And we introduce those things into protoplasm. These are plant cells without cell wall. That's why they are spherical. We can also mix them with gold particles and some other nanoparticles, and we can fire them using the gene gun into our plant material. These are leaf discs of tobacco. As an example, is a bacterial gene that when it breaks down a substrate, it becomes blue. The product is blue. Now, the other hurdle is actually to be able to generate plants from those cells that are transformed. In barley, we use immature embryos, and with hormones and selection, you can generate plants that have or not your mutation. But this process doesn't work in all cultivars or all species, but we have now ways of manipulating that system, so it should work in a lot more species. And once you get the plants, you can then screen them using PCR, polymerase chain reaction, which basically you amplify the gene you want, the area, and you look by sequencing, does it have a mutation or not, and so on. And you confirm that by sequencing. After CRISPR, now I can talk briefly about the chemical mutagenesis, which is TILIN, targeted induced local lesions in genomes, TILIN. We did this in Golden Promise. There are a lot of other populations in different cultivars, but we went this because it's the only cultivar we can transform at the moment. And what we did is basically take the seeds of barley, mix them with the chemical EMS, and it introduce mutations randomly, and we grow the plants. A lot of them will be sterile, and we grow again next generation. Sometimes if it's not enough, we can repeat the process, and we have plants, thousands of plants basically, and we collect their DNA. We set it up in 16 by 16, and we mix all the columns or all the rows. So out of this 96 plates, you'll have 32 samples. And in total, we end up with 768 plants per multi-well plate. And we can screen it for a whole gene that we want, or we can scan multiple genes. We can put 10, 20 genes in one go, and we get mutations. We confirm them by PCR, by sequencing, and we grow this. We go back to the bank and get the seed and grow them, basically. Now, how do we select the genes? Well, I mentioned before that we have these processes, and what we did is we selected a number of genes from different steps. And here is just to show you some of them. All the red genes are involved in different steps. We targeted them using RNAi, gene silencing that I mentioned in the beginning. The blue one, we used CRISPR-Cas9 to knock out completely the gene. And here I'm not going to go through all of them. Some of them are not published yet, some are published. So this is an example of the RNAi silencing. This gene, topoisomerase 2, when you knock it out or knock it down, then you get sterile spikes. This is the control that's fertile, basically. If we target another gene called FANCM with using RNAi, you can see clearly what we want. These are donuts of the wild type, no recombination in the middle. This one is flat, which means they are linked in the middle. That's what we want. Unfortunately, you also get side effects where you have these bridges, fine bridges, meaning basically they are not resolved properly like this. So if you use CRISPR to knock out this gene, what you get, these are the wild type, the two panels above. And these are the two lines of CRISPR. You can see it's completely messed up. So clearly, we need a fine-tuning system because RNAi is low. It's not completely knocked out, but this one is completely knocked out. So we need maybe TILIN mutant, for example, that affect an amino acid, but it leaves the protein still functional and things like that. So in conclusion, we are using multiple omics to understand the dynamic of meiotic gene expression. We would like to increase the data resolution by using single cell RNA sequencing or spatial transcriptome, and that's another powerful tool that has technology that has been developed in the last 5 years. It's amazing. And we managed to manipulate multiple key meiotic genes using CRISPR and other tools. And this toolbox has been very useful for other projects, as I said, in potato and so on. And we targeted various aspects of the cell wall and also pathogens and so on. CRISPR basically is a really powerful tool, and it will be really important for crop improvement. You heard about linkage drug before. That CRISPR now can solve that problem. You can split the bad and the good trait basically in one go. De novo crop domestication, as examples, recently, I'm sure you saw in the newspapers, in the press, the example of tomato or rice. This is a tomato that has been reduced in height, and it makes like grapes basically. And this is good for vertical farming, for example. And it's fascinating. In rice, there is a recent paper showing that you can go back to wild type polyploids like tetraploids and so on. They are good at coping with the environment, but they are difficult to work with to make a new crop from. But CRISPR allowed this Chinese group to actually knock out all the genes like that they don't want, and they end up with a good rice that people can grow, and it's resistant to environmental stress. I showed you only, sorry about that. I showed you before basically the impact of using CRISPR to break DNA, but actually there are lots of functions for CRISPR. And you hear headlines of amateurs can do it. No, it's not true. You need a very good lab, you need expertise, so don't be fooled. And the take-home message, I hope you realize by now that actually there is no such thing as conventional and advanced plant breeding method. For me, it's simply plant breeding methods that are evolving and becoming finer and finer. They are more precise. And with that, I would like to acknowledge all the people, our group in the last 5 years, and the previous collaborators, and I worked at the University of Dundee before. We had a project with these guys for 5 years before I moved on to this project. Also, lots of people from Jamet Hutton Institute doing transgenic plants and sequencing and application in potato and so on. And finally, my collaborators from Stockholm that have developed a new technology of spatial transcriptome. And without funding from all this organization, none of this would have been possible. And with that, I thank you for your attention, and I'm happy to take your questions. And probably I can unshare. So I can, yeah.
Thank you very much, dear Barakate. If you wish, we can move on to the questions on chat.
Anybody?
What manipulation of meiotic crossover established in US in terms of agriculture?
Let me open that. What manipulation of meiotic growth? Not sure I understand the question, but basically we have now some genes that we know we haven't published yet, so that's why I didn't name them, that basically increase recombination in other parts of the chromosomes. And the main problem with all these genes is that if you knock out a gene or you switch it, you knock it down, it tends to affect the fertility. So what we want is plants that are fertile, yet with higher recombination. So we have designed approaches where basically what we call suppressors, where we have a gene that's completely making plants sterile if we knock it down and knock it out. And what happened is when we mutagenize them using the chemical I mentioned earlier, then we can restore what we call suppressors that become fertile because we manipulated another gene. And by manipulating two genes, you get the balance right. So you increase recombination while you keep fertility decent. So I hope I answered that.
Okay.
If I can ask one question.
Yeah.
Are there natural alleles for recombination rates? Because as I found out on last Eucarpia meeting, they found out there are alleles, or at least there is some variability in recombination rates naturally occurring in some populations, right?
Yes, there was a paper in Bali where they showed basically that different land races have different rate of recombination, but that's by simply analyzing the polymorphism and all that stuff. There are other people on maize, for example, who compare basically environment like temperature and things like that, where they show an increase of recombination. So you can, we have done also some work on heat shock, for example. There was a heat shock paper on Arabidopsis recently, but again, heat shock does affect fertility. So, but it's important to understand how those things work to target a specific gene, basically. Ultimately.
But basically, you could be able theoretically to breed for higher recombination rates if you could phenotype for such trait, right?
Yes. Once you find the right mutant or the right gene, yeah.
Okay.
The other question is, what can be the consequences of these genetic manipulations?
As I probably answered that already, is that when you manipulate meiosis, you do get sterility. And ultimately, like we discussed now, what we want is to find ways to manipulate meiosis just transiently, just when you are doing a cross. But after that, you can get rid of the mutation of that gene and so on, because otherwise it's not stable in long term. So yeah, there are consequences of the of this manipulation, and can be minimized once we understand completely the meiosis. It's amazing. The more we learn about meiosis, like everything else, the less we understand.
Yes.
And
thank you.
I have something related with that, maybe. So I remember that the traditional transformation protocols left some marks, like in the DNA, marks of the recombination, like some parts of plasmid DNA and so on. Is this the case with CRISPR?
Yeah, of course, with CRISPR also, if you use DNA, as I said, in the list of way to deliver, but if you use RNA or even protein like Cas9, it can be injected, it can be used in protoplasm, then RNA and protein will be degraded and you have no trace whatsoever.
So basically, we might be even eating CRISPR modified food without knowing it.
You
basically.
could, yeah. I mean, yeah, because even if you did it with a transgene and then you segregate it away, then there's no detectable. The only thing that's there is the SNP that was created. And you could detect the SNP if you know what you're looking for, yes.
Okay.
I mean, just like any other SNP, but you don't know how it was created.
I mean, the DNA is an issue only in crops like potato, for example, where you have high heterozygosity, where if you use seeds for segregating, then you get all kind of shapes. So it's not good for that. So it's better to use RNA or protein.
So
Yeah,
basically,
and that's
it's forbidden in Europe, but it's forbidden only if regulators are unaware.
yeah, but I mean, most companies are not going to risk shipping something, but that's the Calyx soybean that has low the FAD knockouts. Those are on the market in the United States, but they're in a controlled path. So they grow it. They specifically sell it to the farmers that have a specific relationship with crushers, and then they take the oil and they have a specific relationship with the companies that are using their oil, for frying and all. So they keep it that way, but technically in the United States, it's not a regulated product.
Mm-hmm.
And many countries now also are coming out with, and I'll talk a tiny bit about that in my section.
I think
but it's a
in
very
Europe,
interesting topic.
In Europe, ultimately it will come because it will be used around everywhere. Europe can't just, and there is no legitimate reason why not. I mean, I'm happy to ban it completely if it was wrong, but people shouldn't ban it. A technology, it's the product that I agree that for me personally, each potato you make or banana or whatever has to be evaluated on individual, like medicine, but not, because you used this synthetic method, we can't use your medicine. That's, to me, nonsense, but it's politics and
of course,
public and all that. It takes education.
I mean, it's a difference between a product-based regulatory system and a process-based regulatory system, which the US has shifted. It's not completely out of the process-based, but with the most recent rules by the USDA, it's very much product-based now and less process-based. Because USDA, the United States Department of Agriculture, used to regulate anything it was created by agrobacterium, because agrobacterium is a plant pathogen. But if you used a biolistics device and you didn't use any pathogenic sequences, then it technically wasn't regulated by USDA, which made no sense at all.
Yeah.
And so with their new rulings, it actually is very much more focused on what is the product. Is the product something that's already well known out in the field, or is it something completely new? And if it's something that's well known, then actually it goes through very quickly, again, for the United States. But for other countries, every country has its own rules on this, and that's why it takes so long for things to get there. And I'll talk a little bit about that in my section.
Yeah. I think there are.
Okay. The other question is, why is that zygote containing less than the diploid number of chromosomes usually fails to develop, but those with extra chromosomes develop?
Mm-hmm. So basically, when things go wrong, then you get what we call aneuploidy, where you can either have more chromosomes or less. And having less sometimes affect what we call gene dosage. Some genes need to be expressed to certain levels. So if you have only one copy, you will have less expression, less RNA, less protein, for example. But if you have more, it can also be a problem, and it is in trisomy, for example, in humans, and but quite often the balance, and actually a lot of evolution in plant happens through polyploidization, where the genome is duplicated once, twice, three, and you can get even 8 times the number of chromosomes and so on, or even more massive. And that allows all the copies to evolve differently sometimes, or adapt to different environment and so on. But I think that's why basically when you get fertilization, if a zygote has 1 chromosome missing, it's not going to survive.
Thank you. Okay.
Basically, in maize double haploid technology, I think it's similar as in barley and wheat. The plants are, for one part of vegetation period, they are they have only a number of chromosomes. They are haploid. Yeah, they're not. You double the number of chromosomes with the colchicine treatment, but they also survive, but are unfertile as haploids.
Yeah, I think during the polyploidization, either artificial or natural, if it happens in the wild, that's the issue with the meiosis. Quite often they are not stable because meiosis hasn't adapted to segregate all these 6 copies of, like wheat, for example, it's a hexaploid, and it takes time to do that. And when I said earlier about the rice and new domestication, that's what I meant, where basically you can have a tetraploid that has evolved for thousands of years. Why reinvent it? You go to that and you knock out few genes that are bad, like if you want from long rice, you want it dwarf, for example, own, and so on. So you can actually, with CRISPR, adopt tetraploid just like that. Well, if you want to do it artificially, it will be messy for decades before you have something stable.
Thank you. The other question is coming from Sarbesh. What about gene replacement efficiency in CRISPR to improve recombination?
Yeah. So for gene replacement, usually it's done with somatic cells, cells from leaves and so on. And in those cells, actually homologous recombination is very inefficient. What works most is non-homologous end joining, where things are linked together again with mistakes and so on. So we had a project where we tried to increase homologous recombination in tobacco cells by expressing some of the proteins that are involved in that process, some of them from meiosis. Meiosis has high homologous recombination and less non-homologous end joining, for example. And what we are aiming is to use those lines and knock out a gene and try to replace with a fragment we want. Actually, there is a CRISPR-based tool called the prime editing, which can specifically, it's quite efficient, can replace few base pairs, but it's very short. What we want is to be able to replace a whole gene or introduce tags and all that, and that system cannot do that for the moment. So it's an area that I have been interested in for decades now, and CRISPR finally allows us to address that.
Thank you. Is there anybody who wants to ask a question? You can ask any question.
Okay. Well, if nobody else.
No. Yes. Dear Abdullah, thank you for your contributions.
Thank you.
Now we can move on to another speaker, Mr. Alan Wenck. Dear Alan Wenck from the United States of America, working at BASF as head of trade engineering. And now he will give a speech about bringing trades to the world market. Dear participants, Mr. Wenck will answer your questions after finish his speech. You can write your questions to the chat. Welcome, Mr. Wenck.
Yeah, thank you. I will try to share my screen.
You can start.
Yes.
Okay.
Yep, I will try to share my screen and see what happens. And thanks to previous two speakers, you've covered a bit of what I was going to share, but I'll try to get through some of that very quickly then, and
yeah, we can.
Hopefully there's not too much redundance there. Okay, let's see if I can get this on slideshow and if I can grab a pointer or make a pointer. Pointer options. Okay, laser pointer. So yeah, thank you for inviting me to this meeting. I really appreciate it. And I put this together also with the thought that there's a lot of students here. So I also have a little bit about my career path to get to where I am now. And I hope that's not too boring for you all. And I'll talk a little bit about the company I work for and some of the products we have coming forward and some of the technologies we use. So some of the things I'd like to communicate, and I need to hide the thumbnail video because it's in my way, is a little bit about me, like I said, especially since I'm speaking to students, to hear how you get into different career paths, a little bit about the company I work for, and about traits as I see them, and consideration of traits, not only from a scientific point of view. I mean, I work for a company, we need to bring something to market. So there's a lot of considerations when you're in that kind of a field of how you can bring something to the public. It's not it's a bit different than how you bring something out for an academic paper, for example. So it's quite different. And then I'll go through how we discover traits and their implementation. So a little bit about who am I. I started my bachelor. I'm an old guy. You can see the dates. I should have got rid of the dates. I did a bachelor's at a small liberal arts college in North Carolina, United States. And then I went on to do a master's looking at tissue culture responses. So I worked in a lab that had one of the first monocot embryogenic tissue culture systems, and there's genotype dependencies there. And so I looked at endogenous concentrations of hormones and was able to establish that there's a relationship between the endogenous hormones and the tissue culture response. And after that, though, I'd been in school since I was 5 years old. And so I decided I should get out for a while. So I actually got out and worked a bit, and that's why there's a bit of a gap between my master's and PhD. So you don't always have to go straight through. It probably is more efficient if you do, but you don't have to. So I went back to school for my PhD after working a little bit, and I worked actually towards gene targeting in Arabidopsis. This was back in 1996. This was before any of the cool tools that Abdullah was talking about. There was no scissors at that time. And so I failed at gene targeting like a lot of people at that time. And again, it's a good thing as a student to have projects that don't work. I had a bunch of projects in the lab and some worked. I was able to get papers, but actually my main project didn't actually generate gene targeting. And again, I got out and I was working. I got very interested in intellectual property. And so I went back to school for a night school and I did a Juris Doctor, a law degree at North Carolina Central University. They have an evening program. And so I was able to do that from 2009 to 2013. So a little bit different path, but never think you're done learning, never think you're done with education. It might surprise you. Experience, like I said, I got out and I worked between my master's and my PhD. A good bit of that was actually at Clemson University. They have an experiment station where I was working in the field growing growing, scouting, spraying, and weighing. As my boss used to say, we spray them and we weigh them. And that was with the entomology department. And I worked with cotton and soybean and cabbage and collards, and also some other crops. We had watermelon there, we had wheat there, we had peanuts there. But it really helped me get a very good understanding of what a farmer goes through to get a crop to market. It was a university, we didn't have to make money, obviously, but still we were growing, out in the field. We're doing all the work that a farmer does. We're bringing in a yield, and actually things like cotton, we even brought to the cotton gin, and the university was allowed to bring it to the cotton gin and sell it at the cotton gin. So it really gave me a lot of appreciation of what farmers go through and what they need to get a crop out. And it's quite a lot. This was pre-GMOs. So weed control, lepidopteran control were amazingly big topics. And they still are, especially with cotton and soybeans. After my postdoc, or after my PhD, I did a postdoc at NC State working with actually conifers, so tree transformation. And I really started to focus on how do you transform things? How do you put new genes, new traits into plants? This was with conifers, which if you think it's a hard thing to go through a life cycle of a plant to get a result, and that life cycle might be 6 months or something. Conifers, you get results in about 7 or 8 years. So a lot of patience, but the part I worked on as putting new genes into these species actually was quite successful. And I was able to get publications out of that. From there, I went to my first industry job. I joined a company at that time was called Novartis. Now you know it today as Syngenta. And I worked on monocot transformation. So actually I did work at Abdullah. I actually transformed Golden Promise myself. So we developed the agro system in-house for that. I worked with maize, rice, and wheat. And I was very fortunate to be able to get some products that are currently out in the market, including Viptera corn, this is a selfie of me standing actually in a field with Viptera corn, and Enagene corn, which is a thermostable amylase that helps in conversion of corn to ethanol for ethanol production. I also was in charge of a global QC project and a database project. I moved from Syngenta to BASF, where I worked again more in maize and soybean and rice a bit. I worked with, again, I was very fortunate to have a product that's out in the market called Provisia. I'll go over that in a little bit. That was from a tissue culture mutagenesis project. I was also very fortunate to be able to be part of a third-party collaboration where I was able to be in Australia for 2 years working with our collaborators on wheat transformation. That's what's showing some here, some wheat transformants. I left BASF and I joined Bayer Crop Science, where I was head of cell biology and then head of trait validation. And I'll go over what trait validation is a bit more in the talk. So as many of you may have learned or heard, there's been a lot of companies, changes, mergers, and acquisitions. Bayer Crop Science purchased Monsanto. And as part of that purchase, they had to divest all their seeds and traits assets. And I was part of that seeds and traits assets. And so they had to divest it, and BASF actually purchased us. So we were acquired by BASF. So I was in BASF, I left BASF, now I'm back at BASF, and a changing title, a lot of the same kind of activities. So I'm head of trait engineering, and I'll again go over that a little bit. So BASF, it's actually a very large multinational company. It's really largely a chemical company. It's has quite a good sales record of about 59 billion euros and a good profitability of about a bit over 3.5 billion euros. It's extremely large. Like I said, 110,000 employees. It has what they call Verbund sites. That's their big, huge locations, and then other production sites. And we serve customers all over the world. And there's really 6 main divisions, but then there's also corporate research. BASF invests a lot of money in research and development, about 2 billion euros each year. Out of that, actually, the companies that bring in a lot of the money, actually, we in Agricultural Solutions, we have a good share of that investment in R&D. So about 40% of that investment each year is coming to Agricultural Solutions, which I'm part of. But BASF is in everything. It's, gosh, they are in paints on many of your cars. They're in your vitamins. They're in just in batteries. They're just a very diverse company that has activities all over the world in all kinds of different areas. Agricultural Solutions, again, that's the division I'm part of, are part of our shares of sales. So out of that 59 billion, 7.5 billion, or a bit over 7.5 billion euros are coming from Agricultural Solutions. Almost €1 billion in profitability is coming from those solutions. And we're in various parts there also, between breeding, vegetables, seed treatment, chemistry, traditional chemistry, traits, and also digitalization, how do you use digital solutions for a better sustainable agriculture. And just a little bit more, again, within agricultural research, there's about 3,000 people, 26 sites, 4 major research hubs. I'm in one of those research hubs. We deal with a lot of seeds and traits information, but a lot of it's also more traditional chemistry. And also, like I said, digital farming that we're involved in. And really what's the challenge is to feed the world, really. I know that doesn't resonate in social media. It's amazing that when you tell people agriculture is trying to feed 9.7 billion people by 2050, that's not what excites them. But to me, it's an exciting challenge that we need to feed that many people, which is not going to come from increasing land, or where it's coming from increasing land. It's not land we want to be bringing into production. And so we need to have really an increase in productivity of 50%. And that's got to be done without destroying our environment. And so we have various ways of doing this. One of the things that have really helped over years are things like herbicide tolerance traits. I know people say, well, how is that helping sustainability? What it does is it means a farmer doesn't go in and till a field, so the better soil quality. They're allowed to do no-till agriculture has been enabled by herbicide tolerant traits. And we start to couple that with digital farming so that you can say, oh, I don't even have to spray at all, or I spray in very specific spots, again, lowering carbon footprint and impact on the environment of farming. So hopefully by getting a lot of these different technologies together, and there's no one single technology, in my opinion, that will make sustainable farming. It's too complex of an issue for one technology. Again, I'll focus on traits, but it really needs all kinds of different aspects to get there. Some of my personal drivers, there used to be a time when a president would tweet things. Of course, this was before Twitter. One of the third presidents of the United States really was a person who supported agriculture. Y'all are in agriculture. I hope you realize agriculture is our wisest pursuit. Whether it's because most real wealth, good morals, and happiness, I do believe that. And it's a great service you can be doing to your country, and to the world, is bringing a useful plant to its culture. When it comes to my other personal driver, like I said, is coming forward with sustainable solutions. So agriculture has a lower environmental impact. I really recommend you check out Jack Bobo's talk. This is one link to one of the podcasts where you can find his talk, where he has very provocative titled talk called "Can Agriculture Save the Planet Before It Destroys It?" And so he really focuses on what kinds of technologies do you need to really minimize the impact of agriculture on the environment and have a better outcome for society. I saw it said somebody raised a hand. I don't typically mind answering questions in the middle. Just for protocol though, do you want me to start answering questions, moderator?
Yeah, you can do it. Why not? It's okay. Like it's your session and your rules.
Okay, I'm not sure who asked the question. You can use chat, ask questions in advance. Flatco has a YouTube. Sorry, who raised their hand? I saw, again, it just came up on my screen that somebody raised their hand.
I think it was Nikita Richard. I'm sorry if I misspelled.
Yeah, if you have a question you'd like to ask right now, then I'm more than happy to hear it. Okay, I will go on then. But yes, please, if you have a question at the time, I'm okay with you unmuting and stopping me and asking a question. Okay, let's get back into this. Oh, there we go. So ultimately, though, what's the need of a farmer? A day in the life of a farmer is not simple. They have to make amazing amounts of decisions. They have these challenges. Like I said, weeds. I've seen fields that look like this picture right here. This is not a fake picture, especially before some of the weed control solutions we have now. I've seen corn ears that look like this. I've seen fields devastated by insects. So they have multiple challenges. They have weeds to consider, insects to consider, disease to consider, other abiotic factors, nitrogen use, water, fertilizer, all those things. And there's multiple technologies out there, and the farmers they're really actually very wise people. They are familiar with the different technologies. Maybe they don't understand how you get to each one, but they do understand and they learn very quickly how to use it. For example, transgenics in the marketplace have taken off amazingly. To Vlatko's discussion on hybrids in corn, at first farmers weren't sure whether or not they wanted to go with hybrids in corn. Here they have to buy the seed from these seed companies, but that's technology has taken off such that now I don't think there's a corn farmer, a real, other than maybe some subsistence corn farmers who aren't planting hybrids. But then there's also, a farmer has to think of customer acceptance, consumer acceptance. But do they want to go organic because they know they can tap into that consumer market? Do they want to go in a different market that they need to make sure that they can sell their product? And for them, sustainability is really, how to feed a family and deliver a heritage to their son and daughter. I mean, there's a lot of misunderstanding out there that there's these large corporate farms, they're incorporated. So you often make a business. And so technically it's a business, but most of these corporations are held by family farmers. They're not some multinational corporation who owns these farms. It's a family that really actually owns these farms. And so they really do take care of the land and consider how do they pass that land on to their next generation. And they do appreciate the need for options. Like I said, farmers are amazingly willing to look at different ways of doing things. They're very open to that. Somehow I got to get rid of this because it's covering up my top. Oh, I'll have to there's a window that's covering up the very top of my slide, but the one thing, again, as a company, you have to consider the path to market. You can't just say what is the best, most newest technology, what's the coolest way to do it. You have to look at several factors. Can you do it, first of all? The science behind it, the technical aspect is a part of that, yes, but you also have to say, if I bring it to market in this manner, will society accept it? And you also have to look at, am I legally able to bring this to market? So you have to look at the intellectual property landscape. And so, the previous talk about gene editing, indeed, CRISPR-Cas9 is going to be your most easiest and versatile way of doing things. But if anyone has looked at the intellectual property landscape around that, it's to bring something to market may be very difficult. But as an academic institution, to use it as a tool, it's a great tool. And I'm not saying you can't or won't. But to the point of what I mentioned, the Galić soybean, they use Talon technology because it works well for them, and they also have a very strong intellectual property position there. They won't go to CRISPR-Cas because they would have to pay somebody else a licensing fee. So whatever product you're looking at, you've got to look at not just, is it technically feasible? Can I, as a scientist, make it work? But how much is it going to cost me? And often that is going to depend on what is the intellectual property landscape. Do I have to pay a ton of money for different licenses in order to bring this to market? Or, I might go with something a little bit different because I don't have to pay so much intellectual property rights. It may not be the most coolest, most modern science way to get it, but if that's the way I can get there, then that's the way I've got to go in business. And that's the same for multiple products. You have to go through this kind of analysis. That's quite different than analysis you go through in an academic institution. There's things that we drop, and the reason we drop it is not because we can't do it, but technically we can do it. Even maybe intellectual property, with intellectual property we can do it. But if we cannot bring it to the farmer and make it acceptable from a societal standpoint of view, then we can't do that project. It's just something we can't do in industry, because ultimately we have to have a product that we sell. And so that's a big difference between academic and business. I don't mind that. One of my drivers is to always bring things to farmers and to society as a product. And I realize to do that, you have to be able to make a return on investment. So our particular mission in trait research, so I'm in this piece of BASF called trait research, which is within seeds and traits, is to create gene-based solutions to improve food, feed, fiber that benefit farmers and society. And so we work with multiple crops, but primarily wheat, cotton, soybean, and oil seeds. And we have what we call indications, and those are pest control. So things like insects, nematodes, weed control, again, that's pretty self-explanatory, disease control, and what we call crop efficiency. And crop efficiency is things like nitrogen use efficiency, water use efficiency, and the like, and also things like being able to capture carbon better. We have a couple different locations. I'm again located actually in this building right here, although right now I'm working from home. But when I'm not working from home, I am in this building here. We have our greenhouses, and this RTP is Research Triangle Park, North Carolina. And we have 2 large campuses there. And this campus trait research is a small part of that. We're 100 and some odd employees in a facility that has over 700. We have a sister site in Ghent, which is actually in Zwijnaarden, a little technology park outside of Ghent, Belgium. And we also have greenhouse facilities there. BASF has a very large site in Limburgerhof, Germany. Trait research makes a small part of that, but we have some people working there, especially on disease control. So the way I look at solutions, and because the previous speakers actually covered a lot of these, I'll go very quickly, hopefully, through them. So to me, traits solutions can be through conventional breeding, utilizing natural variation. And especially as Vlatko mentioned, you have to identify markers for marker-assisted selection. And hopefully you can get into individual genes as well as superior alleles, because that's the knowledge you need to be able to make very precise modifications. With breeding, as Vlatko mentioned, you bring in a lot of linkage drag. If you can change alleles in a very precise manner, then and you know what those superior alleles are, you can bring conventional breeding. You can speed up conventional breeding quite a bit. Conventional breeding also I consider induced mutation, or variation, mutational breeding, and we'll go over some things on that. The things with conventional breeding, though, you are restricted to variations within the species or crossable, viable cross relatives. It's not something you can just take a trait from somewhere and bring it in. It has to already be within that species. I'll talk a lot about genetically modified, transgenic, bioengineered, whatever you want to use for the terminology there. It still also is using natural variation. The difference is that natural variation is coming from anywhere you can find it. So it doesn't necessarily come, if you're working in soybean, it doesn't have to come from soybean and soybean relatives. It can come from a bacteria, it can come from a fungus, it can come from any source really. And also you can have sort of a similar mutational breeding aspect around there, because once you have that new trait, you can actually tweak it. You can look at directed protein engineering to make very specific changes. And then I'll also talk a little bit about genome editing, which again, you're utilizing induced variation and specific mutations. And the question there, is it really restricted to species? Arguably, how much do you modify a gene before it's no longer what you would consider the gene within that species? But that's more a philosophical discussion than a scientific one. So actually, let's get there. So Vlatko went over very well the classical breeding, and we talked a little bit about mutational breeding, and I'll go into more examples there. And transgenesis. And again, classical breeding, you have long cycles, you have yield drag, unpredictable associations. To break those associations may take a lot of back crosses. And as Abdullah pointed out, sometimes those back crosses, even if they're very tightly linked, it's going to be very difficult to get rid of some of that yield drag to really bring those very specific alleles that you want in. You can speed that up. There's things like speed breeding, where you can adjust lighting conditions. And a company like BASF and all the major agricultural seed companies, we have locations around the world where you can be breeding essentially all year. You don't have to wait for the season in your location. We have breeding facilities in Brazil and Argentina. We have a site in Puerto Rico and of course in the United States, and that's all for soybean. In our wheat business, we have throughout a lot of places in the United States and Canada, and also in Europe for those. And hopefully you can speed things up with mutational breeding that induces things, but again, then you're stuck, you have to make these crosses back. And as was mentioned, when you make mutations, maybe there's a bunch of mutations, some of those are good, some of them are bad. Transgenesis really does allow you to put a very specific trait in, but to Abdullah's point, sometimes your germplasm that you can use for transformation may be not the best germplasm. Again, Golden Promise, for example, for barley is a very old genotype. The same is true like cotton transformation is very genotype dependent. So you want to be able to really expand that if possible. And there are companies out there and there are technologies out there that are expanding the types of the genotypes you can use for transformation. So there's a lot of progress in that field. Vlatko already went over this. I think this is a nice picture though, so I would at least talk for a second on it. Yeah, breeding, 10,000 years of breeding has done a lot for us. We've made amazing progress from something that looked like this, so teosinte, all the way to something that looks like this today. The interesting thing, people say that there's a reduction in varieties or reduction in alleles, but at least what we know of teosinte, there's not so many known varieties. It's not very nice to eat. You have to essentially pound it to get the starch out. Of course, what we have today is corn. You can call it natural corn, but I call it artificial corn. It's really something that is very, very different than its origins. And that's true with all our crops. Wheat is not the same wheat that was developed in Turkey. I mean, that was found there and still is sort of the origin area of wheat, but it's not what was there 10,000 years ago or 15,000 years ago. It's quite different today than what it was. And that's true with all our crops. So mutational breeding is, again, normal breeding really is taking advantage of spontaneous mutations, but you can induce mutations. And again, like Abdullah was saying, you can use chemical agents, you can use physical agents, radiation, biological agents. Actually, that's sort of mutational breeding using things like gene editing tools can fit in there. And they can, mutations can have neutral impacts, positive impacts, and negative impacts. And the problem is when you do like a chemical mutagenesis, as Abdullah was saying, you might have a lot of different mutations and you have to clean it up. And so you might not see positive things because the negative things are knocking things down. It also still has to go through a very long selection process often to get to something that you can bring to market. But mutational breeding also has been very, very fruitful. It's essentially has touched all our plants, especially like ornamentals. You see all these different kind of bulbs with like that previous picture with stripes and things like that. All that's from mutations, induced mutations. But even in barley, what we typically use for barley that's for disease control, MLO, that was a induced mutation, or well, not, it was induced by I believe that was by X-ray induction, but it's still a mutation that is induced by people that gave a disease resistance. So throughout all our our lives, we've been dealing with mutational breeding, and it does amazing things for us. One of the things we've taken advantage of and this is utilizing those scientific knowledge to direct what you're looking for. So we have a collection of EMS mutants as chemical mutagen that essentially saturate the canola genome. And we're looking for traits. One thing we know is canola makes this salique, this fruiting body. When it dries, it pops open and the seed goes everywhere. That's the way canola spreads. Actually, canola is even a man-made crop, but that those types of plants, that's how they spread. Is they have the sleek pops open, the seeds go everywhere. Arabidopsis also has a sleek, and what we learned from Arabidopsis is that you can find certain mutations in Arabidopsis in which that sleek, when it dries, it doesn't pop open. It stays closed. And so we were able to look in our collection for mutations in different genes around this dehiscent zone. And we're able to find in our canola collection mutations in these genes. And we're able to cross those together. And canola is a tetraploid, so you got to realize also you got to find mutations in both sets of genomes. The interesting thing is when we brought the first set of mutations together, indeed, the pods never shattered. But when you ran it through a combine, it also didn't shatter. So the first results and the first attempts were not as favorable as we would have liked. And that's where, you know, back to the point Ablo was talking about, that it may not be just, do you get a knockout mutation? Because maybe the knockout is too much. What is the kind of mutation you need to get to the desired result? And we're able to find actually another mutation that's somewhat leaky. And by combining and looking at which genomes it was even on, by combining those, we're able to make a product that mostly would not open up in the field until the farmer went through with a combine. And with canola, what farmers used to do, and like in Canada, is they would cut the canola a little bit before it was mature. It was called swathing. And then they'd let it dry out because they're worried that if there's a big wind and the sleeks were drying, that the wind would start to make those sleeks shatter. And so that's what they used to do. With the pod shatter trait, they can actually just straight cut and they can get a much better yield. This is showing, you know, a normal swathing. Actually, they don't need to swath, and they do better if they don't swath. So again, it's made a huge impact on carbon footprint, even because swathing means you take a tractor and you run it through a field, and then you have to take the combine and you run it through the field. So you reduce the number of times you run through a field by a whole cycle. And also it gives a lot more flexibility. They're able to let it stand till it's a little bit further mature before they have to harvest it, because they're not afraid that it's going to that the sleeks are going to break open and they're going to lose all their yield. Because once canola seeds are on the ground, you're not going to pick them up. They're too small. It's not practical. Another mutation that we've worked with, and actually this is a product I helped make, is using tissue culture for inducing mutation. One of the things we know when you take things into tissue culture and plant tissue culture, that it seems to accumulate mutations. Some of that's because we know that plant tissue culture actually can activate transposons. And so you get a lot of moving of transposons in tissue culture, but you can also get traits coming out of that. So we were able to take rice, we induced callus, we cultured that callus on a selection media. So this is a herbicide we wanted to see if we could get resistance to. And indeed, after many rounds of selection, we're able to generate individual colonies that when we grow up, we find out that yes, there's a mutation in the gene that gives resistance to this herbicide. And this product now is on the market. And because it's mutational breeding, you don't have to go through many regulatory hurdles. It's actually, back to the question of, is it a process-based regulatory or product-based regulatory? This was a process-based regulatory. Since it was tissue culture mutagenesis, there's not a regulatory regime that you need to go through. And it's something you can bring pretty much every country. again, France is starting to look at some of these things and say, no, 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 but most every other country says, yeah, it's fine. BASF also has a clear field system, which is different herbicide in multiple crops. Again, some of this was by tissue culture mutagenesis, some of this was by chemical mutagenesis. The nice thing with the clear field system, it's a different mode of action. So especially in rice, what they try to do now is ask farmers to use the clear field one year and the Provisia the next year to alternate between what modes of action are out there. Because as you probably know, if you subject a weed species to the same selection year after year after year, it's going to evolve resistance or it's going to die out. And typically weeds are very good at evolving resistance and not dying out. So this is giving a way to manage weed resistance by using multiple modes of action. So now I'll get into our genetically modified transgenesis process. And we really just break that into two pieces, a trait discovery, what we call phase 0, and a trait validation, which we call phase 1. So where do you find new traits? We actually collect environmental samples because in the environment, there's millions of microbes. You can culture thousands of those. We've isolated over 130,000 microbes from the environment. Why microbes in the environment? We know that there's microbes that attack insects. We know that there's microbes that can metabolize herbicides. We know that there's microbes that actually can make things that inhibit other microbes, antibiotic kind of things, but especially antifungals for disease resistant. So that's one area. And we have other sources of getting new genes, but I'll use this example mostly throughout, and also how, especially around insect control traits. So we collect the samples, we isolate individual colonies, we get those sequenced, so we know what we have, and then we start working with those. And the first thing we'll do is grow it up, and we'll take supernatant and we'll put it in insect diet and look for whether or not there's any inhibition of insect growth or insect death. And we always have a control, which is some kind of chemical that's put in that we know will kill the insect. And sometimes you'll find, there's wells here with either very small insects or insects that don't seem to have lived at all. And the different diets, we just have different species of insects that we're looking at. They're the major pests for soybean and cotton, and a lot of them overlap also with corn. So we have this, these strain collection, we optimize fertilization or fermentation conditions, and then we look for an activity, but then we want to know, what is that activity? Is it that the microbe itself is pathogenic to the insect? Does it produce small molecules? A lot of microbes produce complex molecules that can be toxic to the insect directly. For a trait, we need something we can express, which means peptides and proteins. And so we find those usually by traditional biochemistry often. We also do it by genomics. Again, we sequence, and we can look in that sequence and we can say, hey, does it have homology to this certain class of proteins? This is a diagram of the cry proteins, which are crystal proteins that are made by various Bacillus thuringiensis strains. Those are biological control agents that are used, for example, in organic farming, where they take the bacteria the bacteria that's been grown, and they spray it on plants, and those plants then are able to resist the insect. We've been able to isolate from this class quite a bit. You can see all the blue here are things that we've isolated. And the nice thing is these different arms of this wheel often mean different modes of action, often mean different target species, which is really important, because we need to have different modes of action. Again, just as in with weed control, with insecticides, with antibiotics, you give the same thing over again, same mode of action. The thing you're trying to protect against will evolve resistance to that. And so we need to have as many of these modes of action as we can. So we look at that mode of action. We have different ways of doing it, competitive assays. We sometimes know the receptor on the insect and we can look at, is it binding to the same receptor? What is the mode of action? Most of these are pore formers. There are things that insert into a cell membrane, cause it to leak, and that causes it it kills the cell. Again, usually that's because it has this very specific receptor on it and it has to be activated usually by the insect's saliva activates a lot of these proteins. And then, again, we need to evaluate to make sure that we're not putting the same mode of action out because that doesn't do us any good. That we need different modes of action. And so that's once we get to a point where we have something that looks promising, we go ahead and move it into plants. And the reason we do that, we do as much stuff as we can in bacteria first, because those of you students who are working with bacteria, hey, if you're working with E. coli, you can grow it up overnight. You can do an assay the next day. You can make some changes the next day. And you can repeat that cycle very quickly. Once you move into plants, you're looking at things that are going to take a couple of months, even in a good system. And so now I'll move into the trait validation phase 1. As Abdullah mentioned very quickly, yes, we mostly are using agrobacterium. We do use other ways of introducing genes into plants. Most of it's agrobacterium. Again, agrobacterium is this bacteria. It's amazing bacteria that has evolved a way of introducing DNA into plants, and that's part of the way it is a pathogen. It makes this crown gall where the bacteria lives, and it makes the plant make this stuff for it to eat. It's a brilliant bacteria, but it's not so brilliant because we were able to figure out that the bacteria doesn't really know what it's transferring. It's been selected to transfer these things that make this crown gall and make these plants make the food that they're eating. But it's really because there's a sequence here that a protein in the bacteria acts on, and you can replace the sequence that the bacteria normally puts in with anything you want. And then it's no longer a pathogen. Well, it still introduces the DNA into plants, but it doesn't give this response. And it gets incorporated into DNA stably. And then depending on species, you're going to have some kind of co-culture infection media where you mix plant cells and agrobacterium, and then you're going to look at some kind of callus-inducing medium that's going to contain some kind of selection. So you can say, oh, okay, I'm getting these that have probably my new gene of interest in, and then you regenerate plants and do all your analysis from there. And so that's the basic format that we work with. One of my teams is actually a vectoring team, and this is the way a agrobacterium vector looks. You have this right border, again, these sequences that tell the agrobacterium transfer these pieces of this gene piece over into the plant cell. And it has everything you need for doing your standard genetic engineering. A lot of things, even though we use agrobacterium, most of the work is done in E. coli because E. coli is faster and easier to use than agrobacterium. Once we have everything cloned in the E. coli, then we transfer it to agrobacterium, and then there's a selection for the bacteria to maintain that plasmid. And then you have the new trait of interest. And there's key considerations here. And some of these considerations are the same ones you'll have to have for academics, but others are things that you need for industry itself. So of course you need proper regulatory sequences, 5' and so promoters, terminators, often introns will help express in a plant cell. you have to have proper coding for species. Again, DNA is DNA, but a bacteria gene often does not look like what a plant gene looks like. You know, all know about the degeneracy in the code, right? And so the most favorable triplets for amino acid in a soybean gene is not necessarily the same as the most favorable triplet for that amino acid in the bacteria species you isolate it from. So we do recode what we get to make it look like a plant gene, whether it's a soybean gene or a cotton gene or a maize gene or a wheat gene. We recode it to make it look like what should be in that species. And that does help a lot for expression. This is something we do, again, that's not going to be done typically in academics. We do in silico health and safety. And so we want to look early on whether or not this is a potential allergen. And so the way we look at that is we have a database of allergens, known allergens, and then we have a program that goes through an 8-mer sliding scale and looks at potential epitope matches to this known allergen database. We also look at toxin database to make sure it's not a known toxin. We also, one of the big differences we have to do in industry is we try to eliminate open reading frames wherever possible, because when you bring something to market, it needs to be the protein you say it's going to be. If there's other ATGs, AUGs in your whole plasmid here, then those are things that regulatory authorities are going to say, well, how do you know that protein's not being expressed? So as much as possible, we eliminate open reading frames, especially anything in between. Obviously, if you have within the gene AUG, ATG, you can't change that, or sometimes you can, but you don't want to get rid of all your methionines, right? That's essential amino acid. The other thing, again, is to make sure you have freedom to operate components. There's companies that own certain promoters. There's companies that own certain terminators. We need to make sure that we can use the ones that we are using. Our selectable marker, things that we know we can use. Other companies have their own selectable markers. In academics, when I was in academics, I didn't care. I had, 35S promoter. I had kanamycin selection. The fact that at that time they were still under patent protection was not something I considered. But in the United States, actually, even in academics, that's not quite right. But nobody really seems to care. In Europe, there's a lot of places where you have research exemptions. But again, if you're going for a product, something you want to bring to market, then you have to be able to bring it to market. So you either have to have licenses or it has to be things that you have yourself. To put new traits into plants, you have to have, and again, Abdullah went over this a bit with barley. You have to have a robust transformation system. It has to be as quick as reasonable. And again, for plant transformation systems, 2 months that we have currently for our soybean transformation system is amazingly reasonable. And so we've developed this system. Again, it's a system we are allowed to use. We have our own rights to use it, because that's another thing. Again, if you're in industry, you have to consider, can I use this technique? And this is one we can use with our own selection markers. Any system you put together has to have low or no escapes. And those are things that are surviving selection but are not transgenic. Another thing we got to really consider for industry is a good percentage of what's called high quality or registratable events. And what I mean by that is you have to have a large number of single copy. They can't have a lot of other sequences. So if you have the backbone of the agrobacterium, which can get transferred to a plant, in that plant, it's not going to be something regulatory agencies around the world are happy with. And so we try to have systems that generate single copy events that have no backbone in it. And the other thing that is very important, especially again in industry, is having it in a relevant germplasm. Often with soy, it's more also relevant maturity group. So for example, if you're trying to target a pest in Brazil, which we are, you want a very, if you're familiar at all with soybean, you have different maturity groups. You want something in a later maturity group, a 6, 7, 8, because that's what's going to be in Brazil. And if you're going to do field trials in Brazil, it has to be something that performs in that field trial. So you have to have that relevant germplasm around maturity groups. The other thing we do is we do a lot of characterization very quickly in the process. We generate thousands of these plants a year, literally thousands, and we need to weed through those. And so we have a high throughput analysis group. A lot of what we do is really this, the copy number. That's one of the first cutoffs. Again, if it's not single copy, typically we do not move it forward. And that you can find with a very small leaf piece already in tissue culture. You don't have to take it even to the greenhouse. Often then we're going to look at expression. We want to make sure that there's some kind of expression. If we have protein tools, we'll look at protein tools. If we look at, if we don't have protein tools, then we'll look at RNA expression analysis. And again, these things have to be quick, robust, and reliable and high throughput. My teams go through, again, hundreds of these samples a week, all year round pretty much. And so it's something you have to have high throughput systems to get industry type things coming out. We also do greenhouse characterization. One of our traits is, our indications is weed control. So even early on, before we go even into the next generation, we'll often do spray testing and do herbicide damage ratings and only move forward those things that look good. The key considerations when you're in a greenhouse situation, again, in academics also, you need sufficient quantity and quality of seed if you want to do analysis in the next steps. Greenhouse, you have to have good environmental control. For us, stewardship is amazingly important. We cannot have commingling of events. So I have to keep this plant separate from this plant, and they cannot mix. If they mix, then I potentially I can have problems downstream. And so we have a stewardship program that really, we keep everything as separate and uniquely identified as possible. And once you get to this point, you really, again, this might be for an academic, you might grow up the next generation, you do your analysis, you publish your paper, you move on. For us, this is just the first stage in a multi-year, multi-million-dollar endeavor. Okay, and when I say multi-year, it's like 15 years. And when I say multi-million-dollar, it's like $150 million to go from here to product. And this is the kind of things that you see that really, to me, gets very, I get very excited when I see this. Again, I used to work in the fields, and I would see plots where we did not treat, again, there was no GM at that time, no traits out there. But if you did not treat with chemistry, often you would actually have areas that you'd have 100% defoliation. When you have some of these traits in there, all of a sudden you can get to points where there's zero. And again, from here, once you're here, you have to get through regulatory approval from all major and most minor importing and exporting countries. Soybean is something, again, it's grown mostly in the United States, Brazil, Argentina, other places, South America, but it gets exported to the world. It gets exported to Europe in animal feed mostly, it gets exported to China, it gets exported to Japan, Korea. We need to get regulatory approval in each and every one of those countries. If it's going to be grown there, that's one type of regulatory approval. If it's going to just be eaten there, consumed there, we have to get a different kind of approval. And so everything, that's what takes a lot of time and market money to get to market. And these are some examples of what is out there on the market. A lot of these are maize, but also cotton and soybean. And these are things that some of these are no longer even working quite well due to resistance issues, like the Cry1F. This Herculex product is not really efficacious in South America at all anymore. Cry1AB is still fairly efficacious. VIP3A, that's the project I worked on in Syngenta, and that actually is a very highly efficacious protein. Things like herbicide tolerance, glyphosate resistance, Liberty Link, glufosinate resistance, and of course you've probably heard 2,4-D resistance, dicamba resistance. So there's a lot of products actually on the market that are helping farmers. So gene editing. Abdullah did a great job talking about gene editing, so I'll probably go through this very quick. Again, we look at gene editing very much like traditional breeding, but we hope it's cheaper and faster and more precise. So you're able to, if your ultimate thing is to get to an all elite alleles without having to do crosses and back crosses, hopefully gene editing will allow you to make very precise changes to alleles to really speed up making elite material that's going to yield better, have better environmental stress resistance and disease and all resistance. And with gene editing, again, you have various situations, very simple, quote unquote, simple knockouts where you either can make cuts and you drop out something in between, or you make a cut and because you have something cutting there, you just get a mutation. It's, and you essentially are knocking out the gene itself. This typically, again, it's not a regulated material as long as there's not transgenes there. In the United States, it's not a regulated material. In Japan, actually just approved a gene edited tomato, again, as a non-regulated item. There's also, and again, Abdullah talked about precise editing, where you don't let this random process go, you actually make a very specific change. This could be done by various, now there's base editors out there or templates. Again, if you have templates, you have different regulatory concerns than if you don't have DNA and don't have templates. And then you can use editing to stack things together. This then would be typically a transgenic situation, be regulated as a transgenic, because you're putting a gene together in a very specific spot. So really, what do you need? It's really just there's a way you have to have both the targeting, so you have to have that tool, that scissors, as Abdullah was talking about, but you also have a very good way of then finding that target cell, creating that target cell, and bringing it forward. So you have to have not only that piece of that tool, you have to have a way to component delivery. And I use that term component delivery because, again, as Abdullah said, it could be that you're delivering DNA through agrobacterium or biolistics. It could be that you're delivering RNA, or it could be you're delivering, if you're in a CRISPR situation, or not even a CRISPR situation, you could be delivering a protein, some kind of nuclease directly into the cell. It's not, but you have to have a protocol and procedure for doing that. The questions, are you using a repair template or a base editor, all those things. And then how are you going to select something? Say I want to change this allele, but when I change it, it doesn't make any change to the cell. Do I have to put a transgene in there? It's a selectable marker to give me a herbicide resistance so I can say, aha, that's the cell I want. Of course, then when I talk about a regulatory environment, that has different regulatory implications. Now, what I can do sometimes is I can put one editor piece in there and I can put another, and that other editor could be making a mutation in a plant gene that gives you herbicide tolerance. And so you could still use a herbicide selection, and you can hope for both edits in the same cell, which actually can often happen. And so these are things you have to think about. And then, how you've got to have a very robust tissue culture and regeneration system. Again, at least today, it's not you cannot get away from having a very solid cell biology system. I mean, if you cannot transform with DNA, normal transformation, you're not going to get by with doing editing at all. You have to have that level of expertise. And so we've been able to look at this for actually doing a lot of genomics in wheat, part of that for traits, but also for trait discovery. So we also, like in a lot of academic pursuits, we're trying to find out what are the genes, what are the targets. If I drop out this piece of the 5' UTR, do I get upregulation? Do I get downregulation of the gene? Same true on a 3' end. And we've been able to really establish a very high throughput knockout platform for wheat, for example. And you can change things that allow you to do speed breeding, such as flowering time, and that really helps a lot, to speed up what you're working on. And this is an example of actually using a template and an herbicide tolerance. One of the things actually, and this is the exact same herbicide in Provisia, but what we know with Provisia, if you make one mutation in the ACCase gene, you have resistance to a certain class of chemistries that target the ACCase protein. If you can make a double mutation, you're opened up to a whole different class of chemistry. But what we found with normal tissue culture mutation, we still work on it, but it's very difficult to get 2 mutations in the same gene in the same generation. So we did some experiments looking at how to, to use gene editing in a template to specifically make those double mutations, and we're able to do it. But again, to the point, that was asked before, would you be able to know that was transgenic? If there's no other selection in there or no other transgene in there, you would not really be able to know. And indeed, we have used it for stacking. There was a question before, is stacking efficient? There again, it depends. If you have a very efficient transformation system, a very efficient gene delivery system, it can be efficient. So we've been able to show that we can stack against a current product. Essentially, we can stack new genes against a product, and it can be as high as 1 to 5% of all the transformed events that come out are targeted to that stacking site, that stacking location. One thing to be clear, though, that's not always the catch-all for everything. Even if you get to the exact same location in the genome, as was also discussed, you have this packaging of DNA, you have methylation of DNA, you have what's called the epigenome. And what we've seen is even when you insert and it's genotypically identical, the expression's not always the same, and it's usually due to some form of methylation upstream. So it doesn't mean that if you stack in the exact same spot all the time that you're going to get the exact same expression. So that's something you just need to be careful about, is don't assume all edits are going to be the same. And I'm sure we'll see that with knockouts as people start looking at methylation patterns and all that. When you make that cut and you do a knockout, maybe you have some upstream and downstream changes in methylation. It's not, the genome is always a little bit trickier than you'd like it to be. And so there was a question about how precise is it. So actually, gene editing using these tools can be very, very precise. There's papers out there that say, oh, there's a lot of mutations, but really the very controlled studies in plants have shown, yes, there's a lot of mutations, but that's actually, the background mutation rate is amazingly high in plants and actually even in animals. So there's been studies now on clones of cattle. There's been studies on identical twins. The number of SNPs per generation is pretty amazing, single nucleotide polymorphisms. So within a normal non-even gene editing, you're going to have 30 to 50 spontaneous mutations. Tissue culture itself, like I said, induces mutations. And so you can have quite a lot just by the fact you brought it into tissue culture. And back to Vlatko's point, he asked about, does an agrobacterium, it's not always backbone that is causing other mutations. There just seems to be some associated mutations when you have agrobacterium infection, even if it's not. So there's often with these editing tools, if you really rule out the background, you find very few off-target mutations, especially if you're working with a CRISPR system, if you design your guide RNAs correctly. So you can actually get a very high % of target and almost none. And back to the point of mutational breeding, when you're doing conventional EMS, you're generating 18,000 mutations. So it's, the old technologies generate a whole lot of mutations than any new technologies. So just, for reference. And so just in conclusion, I hope I haven't bored you silly, but to me, it's all about how do you get to a better sustainable, higher secure yields. And that can be through traditional breeding, traditional genetically modified approaches, and these new breeding technologies, as whether it's CRISPR-Cas or even zinc fingers or talons. They're all, to me, ways you get into, that you bring new traits to market and hopefully decrease the environmental footprint of farming and increase the sustainability. And that's really done usually by higher and secured yields. And I think that's it. That's where I'm stopping. So I will now try to get back. Wow, chat 14. Wow.
Thank you very much, dear Alan.
Yeah. Okay, let me find where they start. Where they start. Gosh. Okay. This is a link to Jack Bobo's talk. I assume that's where it starts. Was there a question before that? No. Okay, great. Yeah, and I do recommend that. It's great. X-ray is one of the causes of mutation. My question is, why do you use X-rays at hospitals? Is there? Yeah. So it's known. If you don't want to subject yourself to X-rays for more than you need to, right? When you go to a doctor, you don't get X-rays all the time. When your X-ray for mutations in plants, also your timing, how long you're doing the X-rays, the exposure. With medical procedures, they try to limit how much. And the benefit, though, of doing an X-ray in a medical situation is seen as higher than the risk. But can X-rays cause mutations? Yes, indeed they can. I don't concern. I mean, I just came from the dentist last week. I had x-rays for my teeth because it's more beneficial than I worry about the risk. And again, I've not done personally x-ray mutagenesis for crop plants. My understanding is it's much harsher conditions so than you would do for a standard medical x-ray. And with things like EMS, chemical mutagenesis, back to the previous speaker, yes, I mean, you target with EMS almost only a 50% survival rate. If you germinate your things out after EMS treatment and all the seeds pop up really quick, and then you didn't do a very good job. So typically when you're doing mutational breeding, you're really going as harsh as possible without totally killing everything. So it's not that a medical x-ray doesn't have its risk. It's just it's not the conditions that I'd worry about. What do you think about first gene transfer from plant to insect? Oh yeah, that was cool. Would it offer a new way for wow, no, that's a great question. I think it's somehow would it be possible to really direct that? And that's to me the issue is how would you control that? Now, one thing we do, there are strategies of looking at RNAi, for example. So RNAi, insects, nematodes, if they take up a RNA and it's a RNA that is against like a gene in their own body, they actually it can sometimes cause that gene to shut down. It can make a response. And so people are looking at that as a insect control strategy, for example. And also even a sprayable RNA. So you spray the RNA onto the plant and the insect eats the RNA that happens to be on the leaf. First gene transfer plant, would it offer new? Wow, that's interesting. I hadn't thought about that, Ali. That's interesting, very interesting thought.
Alan,
yes.
I can add a comment about that. Yeah, as you said, all about RNAi and stuff like that, but also CRISPR is now used in the gene drive, what's called gene drive,
yes.
where basically you insert a cassette that's lethal in a way in insects, and then you release them, and they will hybridize with other individual, and basically they keep killing especially the females so that they don't propagate. It was considered with a Zika virus, for example. I don't know what happened. So yeah.
Yeah, no, those, yeah, but that's actually engineering the insect itself. Yeah. And that there's actually it's not a gene drive, but there are various engineered insect strategies. So they did release actually engineered mosquitoes to combat Zika, but that wasn't a gene drive. It was a different situation. And there's actually going to be a release of mosquitoes in the Florida Keys, again looking at mosquito control in those areas as alternative to insecticide treatments and such. So I think, yeah, new insect control strategies using genetically modified insects is very interesting. Gene drives, I also have a little bit of concern sometimes about gene drives because, as you say, those are things that can snowball. I've been in different symposiums talking about gene drives, and often they'll have a way to turn off the gene drive. And that is something I think is probably a wise move if you're looking at gene drives. But that's, but yeah, this one about, can you get the plant cell to gene transfer plant inside? That I'll have to think about. Okay, Sharbesh.
Mr. Wenck,
Yes.
there are 3 more questions before this.
Oh, are there? I'm sorry. Okay. Let me scroll
First
back up.
one comes from Richard. How does polyploid are formed?
Sorry.
How does polyploid are formed?
Oh, so that actually probably should go to Abdullah more than myself, but usually polyploids, so I mean, a lot of plant species, tobacco is a tetraploid. Soybean actually is an ancient tetraploid. They're usually a cross that will either spontaneously result with a doubling. When you're talking about that whole canola brassica family, they're all tetraploids, and you can change different genomes by making these crosses. A lot of times you have to go through embryo rescue kind of procedures when you're doing things like that. Wheat itself, right? You have bread wheat, which is a tetraploid, and then you have, or sorry, not bread wheat. You have pasta, which is a tetraploid. Then so durum wheat, and then you have your bread wheats, which are hexaploids. Quite honestly, I'm not the expert on there, so maybe one of the other panelists can answer how you get to a stable polyploid.
Then Alan, maybe it's better if you unshare your presentation.
Oh, sorry. Yeah, sure.
So we can
Okay.
see each other.
Yeah. Yeah.
Yeah.
Yeah.
I can comment on polyploidy. Again, it's about meiosis. So you can cross, as Alan said, two cultivars, or let's say rye with wheat or with barley, and so on. And during recombination, things will go horribly wrong. But there are genes that are known now that you can switch off or replace with a different variant, like some centromeric specific histones, for example, and you end up actually with blocking the division. So instead of having haploid enter pollen, you end up with the embryo sac that develops into a seed, normal without fertilization, for example. But as I said before, that polyploidization is quite often unstable, and it goes through rounds and rounds until you get something like it has been shown that some meiotic genes will be, let's say, if you have 4 copies, they are all expressed. That's not good for the plant. So slowly, somehow, the plant will switch off some of them, and you have only one that's working, and then it becomes stable.
Yeah.
Yeah.
Yeah, I find ploidy actually quite fascinating, but I haven't studied it enough. One thing that always, when people ask me what's one of the most bizarre things that you would consider biotech in plants, I always still say seedless watermelon, because you take a tetraploid and you cross it with a diploid, and you wind up with a triploid that still is viable. It still makes fruit, but then it runs into trouble when it's trying to make seed, and that's how you get your seedless watermelon. But I hope that answered your question. Alperin, could GM crops curb the negative effects of climate change? I'm going to say one provocative answer that I would say they already somewhat are. So herbicide tolerant crops have reduced the carbon footprint of growing soybean, equivalent to like taking 15 million cars off of the road. That's also with insect tolerance, because you're not taking that tractor out and burning diesel fuel to apply insecticide. But to the other point, yes, you could, and people are looking into how do you make plants more heat tolerant? How do you change root architecture, for example, to be able to deal with perhaps drier conditions? Those are things that we look into a little bit, not as much as I'd like, but mostly a lot of the GM climate change issues are also basic stress. So, dealing for plants that can take better heat stress or engineering for better heat tolerance. So I hope that answers your question.
Maybe I have a related question if it's
Go ahead, yeah.
So you mostly talked about qualitative traits, right?
Yes.
These are the like presence, absence, and so on. But what about quantitative traits? And I think students here pursuing the career in quantitative genetics might not be familiar about the long path from gene editing to phenotypes in like oligogenic traits. And
yeah, so
that ends up in genotype times environment interaction, right?
Mm-hmm. Yeah, and so we do have, I did focus pretty much on the insect control story and a little bit on weed control story because they're easier stories to talk about. We do have a crop efficiency group, though, that is looking at different pathways that you need to tweak in order to secure yield under various environmental conditions. And yeah, those are often involving various biochemical pathways that you have to stack together, need to look at multiple locations, multiple environments to really see impact. And they're more also involved in precision field phenotyping, because that's where those kind of technologies come in, is to really be able to see, hopefully early on, to see impact. Back to your point about ultimately it has to be yield, but being able to early predict something that will lead to yield is high value. It's so if you don't have to take everything to a field trial and all the way to a yield trial, then you're saving a lot of time and money, because they're quite difficult.
I agree. Yes. Thanks.
Yeah. Okay. The source of new traits. Okay. Sorry.
This question comes from Eric.
Yeah. Does the source of your new trait driven by market? What are the challenges in the product marketability? Yeah. No. So that is our traits. Again, when you're looking at insect traits or weed control traits, that's driven by the need of farmers for a solution. So when I was working back at the experiment station with cotton, we would spray cotton from June. We'd start spraying all the way until mid-August, or to the end of August, with some kind of synthetic pyrethroid, typically insecticide. After traits, the farmer no longer has to spray except maybe once or twice a season. So that is driven, that's the market was saying we needed this kind of a solution, and the traits then are providing for that solution. The same with weed control. When I worked in Clemson, weeds were an amazing problem. We didn't have traits. A lot of the chemistries were not super good, and you would spend most of your day in the spring on a tractor doing cultivating in a field, mechanical cultivation. So these solutions are driven by a need to control weeds, control insects, control disease. We have an Asian soy rust program, and that's because currently, if you do not protect using chemistry for Asian soy rust in Brazil, you essentially get zero yield or almost no yield. So that's the market driver there. Now, changes in product marketability, that to me, that is where haven't in the past done a very good job at communicating with the public about farming challenges, and solutions, and how those solutions impact the public, especially, as you know, in Europe, GM solutions are non-existent except in Spain with some BT corn, but they're essentially not there. And that's because we did not do a very good job at talking to the public and convincing the public as to the need, and also understanding these traits and how they, how they bring more sustainability to the environment even. Those are messages that I think we are working on doing a better job at, but that is a challenge, is to make sure things we bring to market are things that we can sell and that the market will accept. And I hope that answered your question, Eric. And then, now I jumped down to the one that was already.
Sarbesh has a question.
Yeah, regarding the regulation from USDA for GMOs, for gene edit crops specifically, there could be promoter, some other background vector during agrobacterium. Do they need to remove completely? So the current, Sarbesh, the current rules now, yes. If you have a piece of DNA there that's in your final product, you have to go through some kind of regulatory review. If you used a stable transformation that was expressing, for example, CRISPR-Cas guide RNA, it did that edit, then you segregated it out, you do not have to, it's gone. So there's nothing remaining there. USDA, they don't. And does USDA check for every transgene? So USDA actually they rely on what is submitted. Now, what is submitted, you can say, oh, that doesn't sound very scary or very secure. But when it is submitted from one of our regulatory agencies or our regulatory groups, there is a person in our regulatory group who has to sign their name to it. And I remember the first talk this person gave said, I'm the designated prisoner. If we submit something to the federal government that is not factual, actually it's not only civil liability, but it's also criminal liability. So we do take it very, if we say there's not a transgene there, it's because we've gone through, especially today, it used to be you had to go through and do southern blot, all the vector backbone and pieces here and there, or design lots of PCRs. Now, though, we do a lot of things are fully then gone through with NGS. So we resequence and make sure that there's nothing there that wasn't there to start with. So crossings, giving them a pass. Yeah, again, the USDA though is not going to pull your stuff and do a southern blot or anything else. They have to rely on the submitter to do that. I hope that answers your question, Sarbesh.
Thank you.
Do you produce commercially? Okay. So that's a great question. Resistant to fungal pathogens. We actually do have a ASR, so Asian soybean rust project going on. We have something that's moving into what we call phase 2. And that phase 2 means we think we have something we can bring to commercial, but we don't have it on the field. We don't have it commercially available yet. So we do have native resistance. So of course, plant breeders, there's fungal pathogen resistance out there in the native populations. And so we do have, they always are looking at your common diseases. So all our soybean, there's various native genes that are incorporated to give better disease resistance. Our vegetable people also. But again, those are native genes. They look for, sort of like Vlatko was talking about, they go to wild accessions and various gene banks, and they look for ones that have some kind of native resistance, and then they breed that in. So it's not, right now we don't have a transgenic fungal pathogen resistant crop out there. Again, we're getting closer with Asian soy rust. a background or, sorry, general, is it possible to hide transgenes somehow via background? I
You
don't know
already
what that means.
answered. Yeah, you already answered that question. It's like
Okay, yeah.
part of Sabi's question, but I missed that.
Yeah, yeah, and again, if it really has a transgene in there, no. I mean, there's going to be promoter sequences. There's transgene sequences, and one can question. There was the orange petunia. I don't know if you know the orange petunia story, where there was this orange petunia that was seen growing in Europe, and it was also available in the United States, and a person who was in a lab that was dealing with carotenoid pathways recognized, gee, that orange petunia looks exactly like this orange petunia we made as a transgenic. And lo and behold, when people start looking at it, it actually was transgenic. And then it got pulled from the market. It just now got deregulated in the United States. So I'm looking forward to buying some. But I haven't bought seed yet, but it's now technically it's allowed again in the United States after it was already there and in Europe and lots of other places. But okay, to investigate a gene function, you need to choose mutational insertion lines. Which polymorphism site is preferable to choose to be sure that you will have a knockout plant? Actually, I think the other speakers probably can answer this better. For me, I mean, if you really want to knock out, you can design guide RNAs to the 5', 3', and you can drop the whole gene out. That assures pretty much a knockout.
I can add to that as Alan said, you need to characterize the mutation. It's known now in human cells, for example, haven't been reported in plants yet. When you knock out an exon, for example, actually the cell can skip that exon that's troubled. And so you actually still have a full protein, although you think it's a full knockout. So people need now to start sequencing not just the gene, but also the cDNA, the mRNA basically, to see if you still have a functional protein or not. The other thing is, for example, we have that FuncM gene that I mentioned. Those two mutants, they are not the same knockout. One of them is complete knockout, and the other one is a change of some base pairs in the DNA that actually maintains the sequence and the open reading frame, but by changing a few amino acids in the 5' end or in the N-terminus of the protein, actually it makes the plant semi-sterile. So those are important for academic studies, but for agronomical applications or economical application, you really need to be careful and sequence everything, make sure what's happening before you rush into the market.
And if you're talking also about specific polymorphisms, that's why like work that Vlatko's doing, and we also do a lot internal to really understand what alleles are out there and then understand which alleles are the quote unquote ideal alleles. That is, to me, the biggest challenge now in genome editing. It's knowing exactly what precise changes you might want to make. We need to have that level of knowledge, and we don't really have that level of knowledge.
Because you get proteins that interact with multiple partners, and depending which domain you knock out or which domain you modify, you can lose some of those interactions. The protein is still functional in some pathways, but not others. So yeah, you need to know the gene very, very well, as much as you can before you even start the experiment of CRISPR. Students or postdocs quite often come to me and say, oh, I have this gene, I want to knock it out. First, it's not as easy as they say. You need to think about it in early of your PhD. not last year of your PhD. That's one. Second, you need to know everything that's known to man about your gene before you start. Otherwise, forget it. You can spend an awful lot of time and money for what?
Yeah, yeah, or you have to. So I know like Corteva is systematically going through and chopping things up, but they just have this platform that's amazing, and they have a large group of people that are characterizing all these things. That's the same that they did with they used to have these maize intron lines that are not transposon lines, and they characterized a ton of those just to see what kind of things in the genome, if you tweak, how does it how does it change the plant. But there again, the resource requirements are amazing.
It's probably easier to identify the targets for qualitative traits compared to the quantitative traits.
I think so.
Because generally the paradigm is shifting towards the like infinite number of alleles affecting the scenario dependent alleles affecting the quantitative trait.
We have a comment here about triticale. Yeah, actually it is a synthetic allopolyploid between wheat and rye. That's correct. Again, well, next innovation stage after genome editing stage and plant improvement. I still think we're in this refers back to some of the discussion we're still in a stage of we need better predictive genomics to even get to an after genome editing stage. We need to be able to predict what allele combinations will do. So to me, that's a huge one. In plant improvement, in agriculture and sustainability, digitalization is one I think that is for sure. It's current. It's now. Things like robotics in agriculture are coming. Those aren't in plant improvement, but they're in agriculture and sustainability. I think go ahead.
Sorry, I can add what I mentioned earlier, but just run over, like on a single cell RNA-seq, for example, spatial transcriptome, all what we have been doing so far, it's really, you take tomato fruit or barley seeds, or you grind the whole thing. It has thousands and thousands of different cells, and it makes it very difficult. We rely on bioinformatics, but it's very hard that grinding material together, it averages gene expression in different tissue. So now it is happening. It's starting animal in a biomedical, and actually it will allow the personalized medication, like cancer, for example, and also for agriculture. I think it's the next technology that will be really powerful to understand the mechanism of these genes.
Yeah, it definitely spatial temporal control, because right now a lot of the things we do are very much a big hammer that you slam down and it does for the whole plant. It's not, we're not fine tuning the way we could be fine tuning.
And there is something I want to ask,
sure,
but it's an unethical question, maybe, like, but
I will only give an ethical answer.
Okay, so can I ask, or you are serious?
You can ask. Yeah.
Okay, like I was thinking about the using gene editing technology for bioweapons. Like just imagine plants kill bees in the environment, and the consequences will be disaster, perhaps. Yeah. So do you think, like, it's a general question, is it possible to use plants as bioweapons?
Yes, For sure. I'm sorry. That's not an unethical question. It is a valid question to be asking with all technologies. It's a tool, right? I could make a plant that expresses a toxic protein. You could do that. I could make a plant that we're mentioning things like gene drive. If you were to have, maybe you're a crazy person and you wanted to make a whole species of plant susceptible to a virus, you could probably use a gene drive that would knock out certain plant pathways that make them more susceptible to viruses. Those are things, I mean, it's like anything else. It's, you can use a hammer to build a house, or you can use a hammer to hit someone on the head with.
Yeah.
It's a tool. How you use it and how society uses it is something we have to consider. And that's actually, why, Jennifer Doudna is very, not only a brilliant inventor and Nobel Prize winner, but she thinks about and is trying to organize around what are acceptable uses of this technology that we as a society are okay about, because there's definitely things you can look at and say there's definitely things you could look at and I would say are not beneficial to society, and in the hands of the wrong person, it's that would be, I don't know, I would use plants. I mean, bacteria are much easier often and things like that, but you could, yes. I mean, if you really wanted to.
We do produce exploding corn. It's called popcorn, so
Yeah.
it can pop into your eye.
Yeah. So yeah, any tool and any technology could be used for not good purposes if the wrong people want to do it.
I think in a positive way, there are regulations. People, there are people, crazy people. Look at medicine that we all take. A lot of it is infiltrated by fake or even dangerous medicine. But there are regulations to stop that kind of stuff. So, because ultimately, if you're mad enough, you can design ultimately bacteria or virus that could affect any plant or wipe out rice or maize or whatever you want. But there are regulations and people, it's a bioterrorism basically.
Mm-hmm.
So
yeah, indeed, but it's not what I desire. I just, I'm just curious about it and want to
No,
ask about it.
yeah, and again, to me, you can conceive of things, that. Yeah.
I mean, look at invasive plants. They are not introduced with a bad intention, but just because people brought them from one continent to another as ornamental plants, and next thing they escape, and next thing they become a big environmental problem.
Yeah.
Unfortunately, most of the world still relies on shared moral values. So, despite the political concerns and the political conflicts, I guess this is bioethical question, and most of the world still relies on some shared values rather than some conflicting values.
Yeah.
And also, I put the link for the participation certificate for the participants here. If you like to have your e-certificate, you can just go to the link and fill the form, and it will come to your mail address. And I also want to ask another question about the mindset of the society against the GMO. Like there are such things going around about the anti-pro or anti-GMO or pro-GMO stuff, and like you were working about it in the decades ago, like 2 or 3 decades ago. And do you think the mindset of people has changed in a good way for the anti-GMO or pro-GMO by time?
Gosh, that's a great question. So I've gotten involved in more societal platforms, Twitter, LinkedIn. I give tours. I've talked to regulatory agencies. I talk to different grower groups. And I talk to my neighbors. I see sometimes what I consider positive shifts, and then other times I see things not. I mean, the UK right now is going through, especially with Brexit, is going through a lot of discussions on, well, do we stay with the EU versions of things, or do we look at a different way? There again, to me, I do put some onus back on the people who brought these things forward to start with. It was not communicated well to the public. They did not try to do a lot of outreach. That's where I see gene editing a lot of difference, that researchers in corporations, researchers and academics are being very open about discussions, what it can and cannot do. A lot of the companies that are starting up in this field are being very active about how they communicate, so the public is not caught unaware, surprised, and that they're spoken to in a manner that works. Because again, the messaging in '96 when some of these corn and soy came out was not well done. It was, we're scientists, we know what we're doing, trust us, don't, you have silly concerns. Those were not, that's not the way you communicate to people. And you had, even before corn was such a concern, or soy, you had GM tomato paste sold in the UK, and it was labeled GM and it was sold without a problem. After the communication debacle, maybe I would say debacle, then even those products came off of the market. And there's so many things that are GMO. So of course, insulin, if anyone's out there diabetic taking insulin, that's genetically produced in bacteria. It's no longer gotten from pigs' pancreases. And cheese, if you eat cheese, most of the enzymes that are used to make cheese are recombinant.
And actually, CRISPR was used first in cheese, because
yes,
in some industry, when phage, those viruses I mentioned, affect the bacteria, and that's how they realize that some strains that have CRISPR can actually be resistant. So
yeah, I mean, Rudolf Berengu, that's where his origins sort of are from Danesco, and looking at yogurt and working on strains of bacteria that, because I never thought of it before, but yeah, yogurt manufacturers, they sometimes would get a phage in and wipe out their production, and they were able to understand. Again, they didn't really fully understand the system, but they did get to know the signature molecules. Actually the clustered repeats were known much before anyone knew what they're for. And though they were, Rudolph was one of involved in some of the groups that really started looking at these cluster repeats and using those not only to realize, hey, okay, these kind of strains are resistant against a bunch of bacteriophage, but also to even start doing intellectual property protection. That, our strains have this cluster of repeats, and all of a sudden our competitor has this same cluster repeats, and they know, oh, they're using our strain. and so that's it's an interesting story.
Yeah. And does anyone want to open a discussion from chat in related topics? You can unmute yourselves if you want to say something. Or also you can put your cam on and we can take a picture, maybe.
Yeah.
And let's see. Yeah, face are coming.
Change from pajamas.
Yeah, and we had an issue with the the restriction, because we have limit until 100 people, but we went beyond this and we couldn't accept more. But I record everything and I will upload the recording in our YouTube channel, which will be open very soon. And if anyone here wants to rewatch the sessions, you can just go through and we will send the link to you. And also the chat also, you can see the link for the certificate. And yeah, any discussion can be opened if desired also. But then in 3, okay? 1, 2, 3. Okay, amazing.
Good.
So no discussion from chat, I guess.
Well, I think it's dinner time there. It's lunchtime here. So.
Then I would like to express my sincere thanks to all our speakers and participants. And I would like to thank my teachers, Mehmet Emin Çalışkan and Ufuk Demirel, for their support.
Okay.
Thank you,
Bye-bye.
everyone.
It was very
Bye.
great.
Yeah.
Thank you.
Yeah, I enjoyed it. Thank you very much.
For nice meeting. I am very happy to see all of you in here, and also I want to thank to Abdullah, Alan, and Vlatko for their nice presentation. I'm sure this seminar will be very informative for all agricultural students. Thank you for your contribution.
Yeah, thank
you.
Yeah, I agree.
Thanks for having me.
Without you, we wouldn't make it that much. And thank you so much once again for everyone.
Thanks.
Bye-bye.
Well, take care.
Okay, goodbye. Take care.
Bye.
Bye.
Take care.
See you.
Thanks.
Bye.